So it's my pleasure to introduce Nichole Pedowitz. So Nichole is a native of Maryland and will aggressively defend that state if prompted, and went to the University of Maryland for her undergraduate degree, where she worked in a variety of different labs, most notably the Falvey lab, as well as doing some stints in industry and an REU at NYU, totally publishing 2 papers from her undergraduate days. So that was very impressive. When I was on the USC search committee looking for students, and I aggressively recruited Nichole to come to USC and was lucky enough to be able to get her to join my lab. And her productivity has continued. She's worked on a variety of different projects, ranging from organic synthesis of carbohydrate-based inhibitors to sort of teasing out the cellular biochemistry of the pathway that she's going to talk about today. And I think to date, she has 7 papers from my lab with 2 more in the works. She's about to go off to start her postdoc. And I hope that you enjoy this sort of detective story that Nichole was instrumental in figuring out, that we recently published in Nature Chemical Biology. So take it away, Nichole.
Thank you for that introduction, Matt. Let me share this. And I would like to reiterate what Lisa said. It's so nice to be talking to fellow glycobiologists, glycochemists. It's refreshing because we don't have to kind of convince the world why what we're doing is so interesting. I'm really excited to share with you today what we've learned about how O-GlcNAc modifications of the protein MYPT1 are instrumental in mediating fibroblast contraction. So in our lab, we're interested in studying the post-translational modification, O-GlcNAcylation, which refers to the addition of the monosaccharide N-acetylglucosamine onto serine and threonine residues of intracellular proteins. O-GlcNAcylation is different than other forms of glycosylation in a lot of different ways, one of which is the fact that it's a dynamic modification, so it can be added or removed potentially multiple times throughout a protein's lifespan. And this makes O-GlcNAcylation much more similar to phosphorylation in its role in regulating its protein substrates activity. This dynamic nature is catalyzed through the actions of 2 enzymes, O-GlcNAc transferase to add on the modification and O-GlcNAcase to remove it. And we're really interested in studying this modification due to the fact that it's been shown to be absolutely required for development in Drosophila and cellular survival in mammals, which has a lot to do with the fact that the levels of O-GlcNAc are intimately linked to the metastatic and the metabolic and homeostatic state of the cell. As such, the misregulation of O-GlcNAc has been implicated in a variety of diseases, including cancer, diabetes, and neurodegeneration. The link between O-GlcNAc and metabolism stems from the fact that the majority of the pool of intracellular high-energy sugar donor UDP GlcNAc, that is the substrate for OGT, is synthesized from glucose as an offshoot of the normal glycolysis pathway. So about 2 to 3% of glucose is shuttled through this secondary pathway known as the hexosamine biosynthetic pathway. And you can actually see how changing the concentration of circulating glucose, even in a simple type of experiment like mammalian cell culture, can change the overall profile of O-GlcNAc modification. So shown here are cells that are cultured in 1 versus 9 gram per liter of glucose. And we can visualize how this is affecting the overall intensity or amount of O-GlcNAc modification using this pan-O-GlcNAc antibody RL2. Our lab is a chemical biology lab, so we make and use a lot of different tools to study this modification. And 2 tools that I've used a lot in my research were these inhibitors that were both developed by the Vocadlo lab at Simon Fraser University that globally will affect the amount of O-GlcNAcylation within the cells. We can actually visualize the change that these inhibitors have on the overall profile of O-GlcNAc modifications using the same O-GlcNAc Western blot. And you can see here that treatment with the 5S-GlcNAc, which is a metabolic inhibitor, can completely, in this case, remove O-GlcNAc modifications on cells or almost completely remove it, versus Thymate G, an OGA inhibitor, which increases the amount of overall O-GlcNAc modifications on cells. And the way that we can use these types of molecules to study O-GlcNAc modifications is we can incorporate these to globally affect the amount of O-GlcNAc in cells, and then probe the cells in different ways to look for differences in the phenotypic output between low O-GlcNAc versus endogenous versus high O-GlcNAc modified cells. And this is actually the workflow that we used to sort of discover and then further characterize the project that I'm going to be describing to you guys today. So shown here are 4 videos. On the top are cells that were treated with DMSO, so they have their endogenous O-GlcNAc modification. And on the bottom are cells that are treated with our OGT inhibitor, 5S-GlcNAc. And I should note these are NIH3T3 fibroblast cells, so mouse embryonic fibroblast cells. And as you can see, over the course of this video, the cells that were treated with 5S-GlcNAc and then submitted to a routine media change, so literally just replenishing the media on the cell culture plates, undergo this rapid phenotype over the course of this 10-minute video. So they go from lying flat on the plate, like looking like fibroblasts, to completely balled up and glowy, which was sort of a surprise to us and led us to the question that I'm going to answer today, which is, what is the role of O-GlcNAc in this phenotype? And if you're thinking this question seems very broad, it was very broad at this stage of our project. We really had no idea what was happening other than the fact that changing the media in O-GlcNAc deprived cells led to a striking morphological change. Over the course of sort of ruling out what could possibly be happening, we determined that we were actually hitting on a pathway that's signaled through the signaling lipid sphingosine 1-phosphate shown here. And so here are again 4 videos, and you can see that treatment in O-GlcNAc deprived cells with this signaling lipid recapitulates this striking phenotype that we had seen previously. And so what we wanted to do first was really understand the role of O-GlcNAc modifications on the sensitivity and propensity of the phenotype that we had seen. And so to do this, we submitted our cells to a concentration course of S1P from 50 nanomolar all the way to 5 micromolar. And what you can see in these O-GlcNAc deprived cells is that they are sensitive enough at these low concentrations of S1P to already start to go through a morphological change that just increases as you increase up to 5 micromolar. But what was really exciting about this experiment is if you look at our endogenous O-GlcNAc modified cells, they seem to be relatively resistant to undergoing any sort of morphological change through these low concentrations of S1P. But by the time you get to 500 nanomolar, you start to see some subtle changes. And by 5 micromolar, they look almost indistinguishable from our O-GlcNAc deprived cells, which indicates to us that we are touching on an endogenous pathway that is some way sensitized to the lack of O-GlcNAc modification on one or more protein substrates. And some of these more subtle changes can be visualized using this quantification of the cell area on the plate. We then performed a reciprocal experiment using the inhibitor of thymate G, OGA, or the inhibitor of OGA, thymate G, which would increase the global O-GlcNAc status of O-GlcNAc modified substrates. And you can see here that in this reciprocal experiment, we have a lack of sensitivity where cells treated with concentrations of 5 micromolar still are not quite as sort of changed, affected by the S1P as even the DMSO cells. And so taken together, this really indicated to us that O-GlcNAc was playing a role in the sensitivity in both directions of this phenotype that we were observing. We also wanted to ensure that what we were seeing was sort of physiologically relevant. And so to do this, we cultured our NIH3T3 cells in low concentrations of glucose at 1 gram per liter versus high concentrations at 9 grams per liter. And we were able to see the same effect of S1P in that our 1 gram per liter glucose were more sensitive to undergoing the phenotype. And this increased in sensitivity as you increase the concentration, which was really interesting to show that we know that O-GlcNAc is a nutrient sensor, but we also can tell that it's a nutrient sensor for S1P signaling. So next, we wanted to turn our attention back to S1P. We're O-GlcNAc researchers. We don't really study these signaling lipids, so we had to do a lot of reading. And what we learned was that sphingosine 1-phosphate signals through 5 transmembrane G-coupled protein receptor, titled receptor 1 through S1P receptor 5. And through a series of biochemical experiments, we were able to determine that we were signaling through this second receptor, ultimately sort of funneling through to activate Rho kinase isoforms 1 and 2, which are known to lead to cell contraction and detachment. And when we looked at exactly what substrates Rho kinases ended up phosphorylating, we learned that it's important for phosphorylating these two key enzymes, MLC and MYPT1. When ROC phosphorylates myosin light chain, or MLC, it becomes activated, which allows it to interact with actin, which will lead the cell to a contraction phenotype. MYPT1 is the phosphatase that is known to terminate this pathway. And so an active MYPT1 will dephosphorylate MLC, which allows the cell to remain flat on the plate and prevent contraction from occurring. However, what's really interesting about this pathway is that ROC is also known to phosphorylate MYPT1. And so a phosphorylated MYPT1 is now inactive, which allows MLC to remain activated and push the cell toward contraction. So you can imagine that if ROC is active, it's phosphorylating both MLC and MYPT1, and in both cases, that drives the cell toward contraction. And so the first thing we wanted to test was whether or not we were actually pushing on this pathway, and if this was the pathway that was affected by O-GlcNAc, the change in O-GlcNAcylation that we were sort of forcing on the cells. And so to do this, we performed a phospho time course where we looked at the induction of phosphorylation in both MLC and MYPT1. What we saw in our endogenous O-GlcNAc modified cells treated with this low concentration of S1P is that there's no induction of phosphorylation for either MLC or MYPT1, which is consistent with the fact that we don't see these cells contracting off of the plate. Conversely, if you look at your cells that are treated with our OGT inhibitor and then submitted to treatment with S1P at these different time points, we see a rapid induction of phosphorylation of both substrates, which was a nice validation that this was in fact sort of the pathway that we were ultimately touching on and that was sensitized by O-GlcNAc in some place. And so from here, we wanted to understand which of these substrates O-GlcNAc status could be affecting sort of their role in this triad of signaling cascade. And so both of these substrates, MLC and MYPT1, have been identified in various different proteomics experiments to be O-GlcNAc modified. We've also confirmed that in our own lab. However, MYPT1 has been shown to be constitutively O-GlcNAc modified at multiple different sites. And so for that reason, we decided to move forward and characterize the role that O-GlcNAc modifications on MYPT1 could have on regulating this substrate. So when we look at MYPT1, this is sort of a large scaffolding protein that has multiple different domains. And O-GlcNAc modified regions have been identified at two different parts of this enzyme, one of which are these two sites identified adjacent to the phospho-MLC binding domain. And then there's this second region, a serine and threonine rich region, that have had 9 identified sites directly adjacent to the ROC binding domain. And because of the fact that in our previous phospho-western blot experiment, we had seen that ROC binding was affected by treatment with our O-GlcNAc or OGT inhibitor, we decided to focus our attention on the effect that this sort of very highly O-GlcNAc modified region could have on the activity of this phosphatase enzyme. And so to do this, we generated a series of stable cell lines, we generated a stable cell line where we deleted this domain in a mutant that we called MYPT1 delta. Notably, this cell line was generated using human MYPT1, so we could selectively knock down endogenous mouse MYPT1 in all of our subsequent experiments that I'm going to talk about in the next couple of slides. But what we wanted to do first was determine whether or not we had in fact sort of removed the majority of the O-GlcNAc sites on this protein. And to do this, we employed a mass shifting assay that was developed by Linda Shea Wilson at Caltech. And the way that this assay works is essentially you're incorporating a mass tag to all of the endogenous O-GlcNAc modified sites on an enzyme, and then you can sort of see an overall upward shift, which is consistent with the number of sites that there could be. And if you look at this result, you can see, especially in this 2KD mass tag, that the overall number of O-GlcNAc modified sites on MYPT1 delta is much decreased, which is consistent with the fact that we deleted most of the sites, but not all of them. And so we moved forward sort of validating the fact that these were in fact bona fide O-GlcNAc modified sites. The first experiment that we did was to determine whether or not these cells, the MYPT1 delta cells, were just as sensitive to treatment with this concentration course of S1P as our previous cells that we had treated with our OGT inhibitor. And what we saw was that MYPT1 delta cells treated with this low concentration of S1P are already starting to become sensitized and contracting off of the plate. And this just increases as you increase the concentration of S1P. Conversely, cells that are stably expressing MYPT1 wild type are just as resistant as the previous sort of DMSO treated cells, which is consistent with the fact that our model is saying these sites on this protein are what are important for regulating the process that we had been visualizing. So at this point, we have this model in mind where MYPT1 is O-GlcNAc modified. And when this is the case, ROC is somehow not able to bind or phosphorylate the enzyme as effectively. However, when you delete these modifications, in the case of this mutant, literally delete them, you are able to more readily have ROC binding, leading to an increase in phosphorylation, which inactivates the enzyme and allows MLC to become phosphorylated. And so to test whether or not this model was what we were actually affecting, we performed the same phospho-MLC time course or time course on MLC and MYPT1 that I previously described. And we saw that in MYPT1 wild type cells, there was little to no induction of phosphorylation, consistent with them staying flat on the plate at treatments of this low 100 nanomolar concentration of S1P. However, this was super gratifying. MYPT1 delta cells treated with this same low concentration of S1P had a rapid induction of phosphorylation, which is, again, consistent with the fact that we saw them contracting and also supports this model that we have in place about these sites being what's really regulating this triad of proteins. What we wanted to do next was understand whether or not the sites that we had deleted were really what was regulating this process, or if there could be other O-GlcNAc modified proteins that we hadn't been considering that could be also leading to some amount of regulation of this process. And so to do this, we performed an experiment where we took MYPT1 wild type treated with our global inhibitor, 5S-GlcNAc, versus MYPT1 delta cells. And what we were hoping to see is that MYPT1 treated with this global inhibitor versus delta would have the same extent of contraction, and that would sort of prove to us that the majority of the what we were seeing was occurring because of modifications on this region. And that's exactly what we saw. That might be more clearly visualized if we look at our quantification of cell area with this red and orange bar both being consistently lowered, which is consistent with contraction occurring at the same rate in these two different conditions. And so at this point, we feel like we've bridged some key observations in the field of O-GlcNAc and also potentially in the field of diabetes. These observations are that O-GlcNAc is globally increased in diabetes. I mean, there's just more circulating glucose, which makes sense if it's being shuttled through the hexosamine biosynthetic pathway. We also know that S1P promotes diabetic wound healing, and that fibroblasts are crucial components of the wound healing process because they're contraction and migration literally pull wounds back together. And so at this point, we sort of have this model where we think that because glucose and then subsequently GlcNAc is elevated in diabetic patients, MYPT1 is more readily O-GlcNAc modified, which leads to an inhibition of S1P signaling in diabetic patients. And so to test whether or not sort of this was sort of relevant in the context of fibroblasts in an in vivo setting, we decided to use a three-dimensional collagen matrix assay that's been shown to recapitulate the way that fibroblasts act in vivo, but in a in vitro cell culture dish. The way that this assay works is we take dermal primary fibroblasts and we mix them with collagen, allow this mixture to polymerize, and then let stress form over 48 hours. We can then release our little jello fibroblasts, gelatins, and add a contraction factor, and then measure the diameter change. So an increase in diameter change means that there is more contraction. When we tested this in our endogenous O-GlcNAc modified cells versus those treated with an OGT inhibitor, we saw that there was in fact an increase in the diameter change in our low O-GlcNAc condition, which is consistent with the idea that O-GlcNAc inhibits dermal fibroblast contraction. So when you actually have O-GlcNAc, they're less able to contract. We can also see this in a concentration course of S1P. It's consistent that DMSO treated cells are more inhibited and less likely to undergo diameter change, meaning that they're less likely to contract. So at this point in the project, we're really excited about what we know so far. We feel like we've characterized S1P signaling and how it mediates modifications on MYPT1. But moving forward, what we're really interested in studying is what is the specific mechanism by which O-GlcNAc inhibits MYPT1 phosphorylation. We have some preliminary data or some preliminary experiments that have shown that in 5S-GlcNAc treated cells, there is more MYPT1 ROCK binding even before treatment with a signaling molecule like S1P. So we think that it might be related to the ability for ROCK and MYPT1 to literally interact. We're also interested in understanding what other proteins and pathways could be contributing to the phenotype that we're seeing. And ultimately, what we're really driven by is understanding how O-GlcNAc modifications of MYPT1 can affect wound healing in diabetic patients. And so with that, I would like to thank a lot of the people that have helped sort of with this project. I would like to thank Anna and Narek, who were both instrumental in training me for this project, and Anna, who started it with me and really set the groundwork for the discoveries that we were able to make. I want to thank Moira for joining the project, and we've sort of, since this paper has come out, also characterize MYPT1 signaling with a different lipid lysophosphatidic acid. So she's been it's really exciting to train someone who's going to take on some of these larger goals for the project. And obviously Matt for being my mentor, always supporting me, and all of you for listening today. I'm really excited to answer any questions that you might have or any comments about the work that I just described to you. And thank you all.